When you look at different organisms, there's a variety of different sexual life cycles. So we're going to compare the fungi to animals and plants to see how the timing of fertilization and how meiosis can vary. So no matter what we're looking at, though, the three types of life cycles are going to share important results. There's always going to be chromosomes being cut in half, and once they're cut in half, you're then going to double. There's going to be doubling and halving that are going to go back and forth, and that's going to contribute to the genetic variation that you get with sexual reproduction. So we'll start with animals. When you go through animal sexual reproduction, there's going to be fertilization where there's a sperm cell and an egg cell. So we could say that's the egg, we could say that's the sperm. They're going to come together through fertilization, and they're going to make a zygote. So N plus N equals the 2N. So that's the diploid cell. Now that diploid cell is going to go through mitosis and turn into a multicellular organism. So that's the multicellular organism like we are. Once that thing matures, it's going to use meiosis to then make sex cells. So they're going to make sperm and egg, and once again, sperm and egg come back together. So the gametes are the haploid cells, and they stay unicellular. The diploid cells are accomplished through mitosis, and they're going to be multicellular. Now, when you look at the plants and algae, they're going to also do this back and forth. They're going to have the sex cell, sperm and egg, come together through fertilization to make a zygote. That zygote's going to go through mitosis to make a multicellular organism. That multicellular organism is going to produce, once again, these haploid cells through meiosis that are called spores. You don't have to necessarily know that. But they are then going to make the sex cells. But the key here is that the haploid cells are multicellular and the diploid cells are multicellular. When we looked at animals, only the diploid cells were multicellular. In plants, it's both haploid and diploid cells that are multicellular. And then in our last group, the fungi, once again, we have the haploid and diploid stage. The haploid stage is multicellular. That's going to produce sex cells that come together into a zygote. However, that zygote stays unicellular, and then it makes these sex cells that are haploid once again. So here, we have something completely different. So in fungi, the 2N is unicellular. In plants, so that's fungi. In plants, both 2N and N are multicellular. And in animals, it's the 2N that's multicellular. And the N is unicellular. So that's the key to understand there. When we look at the haploid and diploid stage, haploid stage is always made by meiosis. The diploid stage is made by those sex cells coming together through fertilization. The question is, where's the multicellular stage? In fungus, it's the haploid cells that are multicellular. In plants, it's haploid and diploid that are multicellular. And in animals, it's the diploid stage that's multicellular. So based on what you just saw, you should be able to answer these questions. If you're doing mitosis, can haploid and diploid cells both go through mitosis? And the answer to that is yes. If you have one cell turns into two cells, it doesn't matter if that's an N cell to make these or a 2N cell to make these. That can happen in both cases. You just saw in plants both haploid and diploid stages could be multicellular, which means they went through mitosis. So the next question, can haploid and diploid cells both go through meiosis? Well, meiosis, remember, is a reduction division. So that means you reduce the chromosome count by a half. So if you had a human cell, for instance, that's 46 chromosomes, that could turn into a sex cell with 23 chromosomes. So yes, that can work because that's a diploid cell. But what if you had a haploid cell, a cell with 23 chromosomes? Could you go through meiosis to make 11 and a half chromosomes? No. So haploid cells cannot go through meiosis, diploid cells can. So when you look at the process of meiosis, you're going to have to know the general principle here. The good news is you're not going to have to know the individual steps. Remember, in mitosis, you had to learn prophase, metaphase, anaphase, telophase. In meiosis, I just want you to know the overall process of what's happening. So in meiosis, you're going to have one cell, and then we'll use humans for an example. If we have that one cell with 46 chromosomes, that's going to divide by meiosis 1 into two cells, and these have 23 chromosomes. But it's not done. They will both divide again into 4 cells, and they all have 23 chromosomes. And this one is called meiosis 2. So there's meiosis 1 and meiosis 2. And in the end, you get 4 cells, 1, 2, 3, 4, with half the number of chromosomes, 46. Half of that is 23. So there's 23 here, 23 here, 23 here. That's meiosis. Compared to mitosis, remember mitosis, you start with one cell with 46, and out of that you get 2 cells with 46. So this is mitosis, this is meiosis. So if we have a cell that's diploid, 2 homologous chromosomes in it, when you do meiosis, you're going to split the homologous chromosomes apart. So one will go here, one will go here, and then in meiosis 2, you split the sister chromatids apart. So one goes there, one there, same thing here, one goes there, one there. So you started with a cell that's diploid with 2 chromosomes, and in the end you got 4 cells that are haploid, each with 1 in. For this case, if this was humans, it would go from 46 chromosomes to 23 chromosomes. So as I said, you don't have to know the individual steps, but I will highlight something here. So looking at meiosis, we'll do meiosis 1, and what's going to happen is the chromosomes condense, chromosomes line up, but here's where it's different. Remember in metaphase of mitosis, when you looked at the chromosomes, what they did is they lined up single file across the cell. So whether you want to look at it like that or whether you want to look at it like that, they just lined up single file across that cell. However, in meiosis, something different happens. In meiosis, what they do is they find their homologous chromosome partner. So remember, homologous chromosomes are chromosomes that carry the same information. One came from the mother, one came from the father. So in this case, you have 46 total chromosomes, if this was a human, but there's 23 on this side and 23 on this side. So then when you split them apart, 23 go that way, 23 go that way. That's how we get half the number. But you don't have to know the names of these phases like you did mitosis. And then it goes into meiosis 2, chromosomes condense. This time those 23 line up single file, and then we separate the sister chromatids. So if this was human, there's 23 in a single line. Now there's 23 going that way, 23 going that way. So that's in the end how you get 2 cells there with 23 and 2 cells here with 23. So you get 4 cells with 23 chromosomes. That's what you end up with at the end of meiosis in humans. So if we compare these, mitosis has 1 division. 1 cell, 46 chromosomes, into 2 cells with 46 chromosomes. They stay diploid, double the number of chromosomes. In this case, this term synapsis does not occur. So remember when you did mitosis and you looked at metaphase, the chromosomes lined up single file, right down the middle of the cell. However, here's what's different. In meiosis, when those chromosomes line up, they find the homologous chromosome partner. So whatever ones match, they find their partner and go together. So synapse is a junction. So what they do is they join by finding their homologous partner. So that's what I mean by synapse. That does not occur in mitosis, but it does occur in meiosis. And then, of course, meiosis has 2 divisions, and you saw those 2 divisions where you went from 146 to 2 23s to 4 23s at the end. And they're haploid cells because you started with a diploid cell and then you went to 4 haploid cells. And the way you get it to split in half is that you find the homologous partner, you join them, and then you split them. So that's what synapsis means. So visually, chromosomes, they condense. During metaphase, they line up single file. Meiosis, chromosomes condense. During metaphase 1, they line up as homologous partners. So when these ones over here split, they split with even numbers going both directions. So you still get to 2 diploid cells in the end. But over here, since they're lining them up as partners, you get half of them going that way, half going that way. Then they divide again, so that's why you get 4 haploid cells in the end. So the importance to understanding all this is getting genetic variation. That genetic variation is going to be vital for healthy populations. So as we look at this idea of sexual reproduction leading to genetic variation, there's going to be 3 mechanisms that really are responsible for creating that genetic variation. And I'm going to go over these individually. You have independent assortment, crossing over, and random fertilization. If we start with independent assortment, what this is referring to is that the maternal or paternal, which means mother's or father's chromosomes, can be oriented on either side. So remember in meiosis, the chromosomes line up as their homologous pairs. So they line up matched with their pairs like this. Well, if I say that this green is the mother and this, it's not showing up, this green is the father, well, that's one scenario. We could have the mother on this side and we could have the father on this side. But couldn't you also have mother on one side, father over here, and mother over here? So the way these two line up have no impact on the way these two line up. If they want to line up here, left or right, well, they can line up here, left or right or left. It doesn't matter. So they sort independently. So that's what independent assortment means. When you have those chromosomes, the way one partner pair line up has no impact on the way the rest of them line up. Visually, we could have a possibility here where the father, which is blue, the mother, which is red, you could have them both on the same side, and then the mothers both on the same side. And that would lead to this combination of sex cells down here. But there's another possibility. You could have a father-mother and a mother-father on opposite sides. Well, that would lead to this combination down here in the sex cells. So you see there's different possibilities of what the end result could be. That leads to genetic variation, and that's called independent assortment. The way these two line up here don't impact how these line up. If these line up like that, they don't care. They could line up in the opposite direction. You're not going to have to do the math for this, but I do want to show you the genetic variation that results of this. Now, when you look at the number of different possibilities, it's 2 raised to the nth power. And that n depends on what the haploid number for the organism is. So for humans, the haploid number is 23. So if we take 2 raised to the 23rd power, that results in 8 million different combinations, meaning that in sperm or egg, there's 8 million different possibilities of the genetic arrangement they could have inside of them. Now, another thing that happens is when you get those homologous chromosomes lining up in that synapse, remember the synapse was the junction, they get really close. And the other thing you have to imagine is that these chromosome arms are not stiff and rigid. These chromosome arms are just kind of flopping around out there. And if they get close enough as they're flopping together, when they touch, they can actually crisscross each other. So that's called crossing over. So here you see the paternal, the father's on one side, and the maternal, the mother's on the other side. And when they get close enough together and they touch, you can get little bits and pieces mixed up on opposite sides. So once again, that crossing over leads to more genetic variation. The thing about this is it's completely random. This happens all the time in chromosomes, but it's not something that you can necessarily put a calculation or a prediction on, which leads to genetic variation. It's basically endless genetic variation. And then the last point is random fertilization. So remember that during fertilization, you have two sex cells coming together, a sperm and an egg. So it only takes one sperm cell to fertilize that egg. The question is, which sperm cell gets to which egg? Remember, based on the first principle we talked about, independent assortment, there's 8 million possible combinations that you can find in a gamete. So you could have a sperm with 8 million combinations and an egg with 8 million combinations. So if you take the probability of those 8 million multiplied by the factor of those 8 million, you're going to have over 64 trillion different possibilities in the genetic outcome of that individual. And that's why that even though you got your genetic makeup from your mother and your father, you're not genetically identical to them. And even if you have a sibling, you're not genetically identical to them, unless, of course, there's identical twins. That's a completely separate story. However, you're a unique individual, and nobody's going to have your exact genes that you have. There's endless combinations. Just based on 2, it's 64 trillion. Throw in there crossing over, it's endless. So why is that genetic variation so significant? It's because of natural selection. When you have organisms that are genetically different, there's going to be some that are better adapted for the environment they live in and some not so adapted. Those ones better adapted are more likely to survive. But since the environment's never constant, the environment's always changing, all this genetic variation helps to ensure that populations continue on over time. So the significance of that sexual reproduction leading genetic variation is survival of the fittest. It's natural selection.